Yeah. I'm gonna... Now you're seeing, right? Yeah. So, hello and welcome from others. We welcome you here. We are organizing for the first time at the University of Lausanne the 3-minute thesis competition, which is the competition that we ask fellow PhD students to present their thesis to the general audience in 3 minutes in English. And we are here live from Genopod, and we are also live on YouTube, and thank you for joining. So at the beginning, I want to give you an overview of the competition. The 3-minute thesis competition is an official competition, first organized from the University of Queensland for PhD students, and it gives them the challenge to consolidate their ideas and research discoveries so they can be presented concisely to a non-specialist audience. This has traveled worldwide, and this is the first time we're trying at the University of Lausanne to bring this competition for all the PhD students of our faculty. Then the rules, to explain them to you, each participant is allowed to have static...
I was talking with something. I managed to connect it.
Hello guys, if you can please close the microphones.
Okay.
The VAR platform.
Yes.
It's the COVID year, so we have technicalities, but we're fixing. Anyway, so each participant is allowed to have a static PowerPoint slide, and no transitions or animations are allowed. They're not allowed to have any other electronic media, like music and videos. They're not allowed to have props to hold anything that would help them to present easier. No music and no poems or anything. They're not allowed to exceed the 3 minutes. Everybody who exceeds the 3 minutes is disqualified, and we are taking account the time. And yes, so then we will have the jury who will decide about which participant did their job better. So the judging criteria, they go on the comprehension and content, because this is what they're judged. And they have to be able to provide to explain their research with by understanding the background and significance of their research, to give the presentation clearly describe and their impact, while to show us how a clear and logical sequence, what was the topics of the research significance, and did the presenter spend adequate time on each element of their presentation, or they elaborate for too long, like I'm doing now, or the presentation was rushed. Then we also have to judge the engagement and communication, how much they engage the audience, which is now the audience is virtual, that makes their job more difficult. And if they trivialize their knowledge, if they convey their enthusiasm, because what we're mainly looking is the enthusiasm to capture and maintain the audience attention, to have sufficient stage present. Again, please don't be strict with them, because some are going through Zoom. It's a difficult time, and the eye contact is difficult at the moment. And whether their PowerPoint slide has helped them, was it clear and legible and concise. However, please take into account that the PowerPoint slide is not mandatory. Some participants choose to not use them. So then it's a moment to introduce to you our judging panel. We have with us Professor Alexandre Roulin, who is Vice Dean Adjoint from the Department of Ecology and Evolution. We also have Katarina Zizarova and Dmitry Kopelyansky. I'm sorry, Dmitry, if I mispronounced. They are former FameLab winners of Switzerland, and we're happy to have them with us because they know how to communicate with the public, and they're here to give their feedback to the participants of our event. So starting, I want to tell you that we have 7 participants today. The first 3, they go through Zoom, and the next 4 are gonna be presenting here live to us because of COVID rules. So to start, I will ask Afonso to connect and present to us.
Yeah, I'm here.
Yes, just
Can you hear me?
Yes, I'm gonna mute us, Afonso, just a moment so that you can have your video.
Sorry. Okay. Am I allowed to start now, or how does that work?
Afonso, when I tell you, I guess that Dimitra from the other room is taking the time and you can start. So, or Dimitra, can you hear us? Can you start, Afonso?
Yes, when you say go, I will also start the timer.
Okay, so go.
As you are probably aware, we humans are never alone. We permanently share our body with hundreds of different life forms, and among them, we have bacteria. Most bacteria coexist with us, not causing any symptoms or disease, so their impact seems inconsequential to us most of the time. However, if you look at things from the bacterial perspective, you'll find a world that is dominated by war, by the struggle to survive, and by being the fittest. For example, when you look at the human nasopharynx, or upper respiratory tract, we find it to be inhabited by hundreds of bacterial species. One of such species, and one of the most prevalent, is Streptococcus pneumonia, which, depending on the circumstances, can either cause disease or peacefully coexist with us. This is especially true on the young and on the elderly. As you probably are aware of it, Streptococcus does not live alone with us. It needs to interact and compete with all the other bacteria that live in the same environment, the upper respiratory tract. The study of these interactions and how Streptococcus pneumonia survives and even thrives in these environments is the current focus of my PhD. The question is then, how do you approach this problem? Well, one way is to look at all the genes of Streptococcus pneumonia and test which ones allow the bacteria to survive or not in the presence of other competing bacteria. For example, imagine you put together Streptococcus pneumonia and Staphylococcus aureus, which is another bacterium that often coexists with us, and that in these circumstances, Streptococcus seems to survive relatively well and actually puts a fight against Staphylococcus. Now, on another scenario, imagine you eliminate or hinder a gene, let's call it gene A in Streptococcus, and perform the exact same experiment. However, this time, you see that Streptococcus has a lot more difficulty surviving in the presence of Staphylococcus. Now, I think we can all agree that the obvious conclusion is that gene A, therefore, seems important for the Streptococcus survival when in the presence of Staphylococcus. And this is exactly what we just did, but for all the Streptococcus genes, using a technique called CRISPR-Seq. And what we found is that there are indeed a few genes that seem to be required by Streptococcus to fight, compete, and even survive better in the presence of other bacteria. And one of such genes is quite interesting, as it seems to be involved in degrading proteins outside the cell. One could then envision such a gene as being able to degrade the surface of competing bacteria, therefore giving Streptococcus the competitive edge over the enemy, Staphylococcus aureus in this case. With this talk, I hope I interested you a little bit more on the perspective of bacteria have on other bacteria, and in a way isn't much different from the one we have. Thank you.
Thank you, Afonso. And within time, so I move to the next participant. There is Elodie Huber, if I pronounce correctly. Sorry again. When I say go, Dimitra, you start counting, right?
Okay.
So go.
What is something we can pretty much all agree to be interested in? Death. You're correct. Well, in Switzerland, cardiovascular diseases are the main cause of death for both men and women. What is something you can't say you're not interested in? Sex-related disparities. Correct again. While studies in the domain actually show that mortality and recurrence after an ischemic heart event are more frequent in women. These facts are something that the Anglo-Saxon literature has shown many times and for big populations. However, in Switzerland, the biggest population for a 10-year study was around 29,000 participants, whereas the official numbers, according to the Federal Statistical Office, should be around 175,000 patients for that period. It is then hard to get a clear picture of a problem with only 16% of all the cases. But there's a solution to that. Hospital administrative health data. We take a minimum set of data that all places of care must collect for each admission. With these, we were able to extract all patients admitted for ischemic heart events in Switzerland between 2009 and 2017. This brought us to 224,172 participants, where 32.5% were women. Now I know what you're going to tell me, that I can't compare what's incomparable, because you know, as trivia winners, that women with ischemic heart events are on average older than men, and that women tend to have more unstable angina and more non-ST segment elevation myocardial infarction, NSTEMI for friends. Whereas men tend to have more ST segment elevation myocardial infarction. That's STEMI, the bigger brother of NSTEMI. And you're partially right, because these two facts were also found in our study. However, even with these factors considered and many other potential confounding factors like nationality, type of insurance, and mental and physical comorbidities, I am sad to announce you that in the adjusted models, women were still less likely to undergo the right investigation and receive the right treatment compared to men. One reassuring outcome was that women were more likely to die in the unadjusted models, but not in the adjusted models. This outcome was more correlated to the bigger age. However, even if the difference in management did not impact directly on mortality, it did on morbidity, because women were more likely to have complications from the hospital care, they were more likely to have hospitalizations longer than 5 days, and they were more likely to be readmitted. Therefore, efforts must be carried out in order to correct this and offer men and women fair and quality healthcare. Thank you very much.
Thank you, Elodie. So we're going to take all the questions at the end of this section. For Afonso, Elodie, and Julian, at the end of the section, we're going to ask our jury if they have questions for you. Okay, so then when I say go, Julian, you're
okay.
next.
You can hold, you hear me? It's okay, yeah.
Yes, so go.
We have plenty of bacteria that reside in the nasopharynx. Most of the time, these bacteria are commensal, playing an important role in the defense against respiratory pathogens by acting on our immune response and competing with pathogens. However, what you may not know is that some of these commensal bacteria can become pathogenic if the right conditions are met. For example, Streptococcus pneumoniae is an opportunistic human pathogen and can lead to pneumonia, meningitis, or even sepsis in the worst cases. It is responsible for approximately 1 million deaths per year, and it is particularly abundant in immunodeficient people, the elderly, and children. With the rise of multi-drug resistant strains of Streptococcus pneumoniae, the need for new therapies is becoming more and more urgent. It is therefore key to understand how Streptococcus pneumoniae behaves, divides, and conquers the immune system. Extensive work has already been performed over the last few years to precisely define the sequence of events leading to a perfectly shaped and divided cells. However, as the cell division mechanism of the pneumococcus is very atypical compared to other types of bacteria, a lot of genes involved in this mechanism still have either an unknown function or are not yet fully characterized. Consequently, to find new genes that are involved in cell division, I established a new kind of screening in the pneumococcus. Commonly, a screen tests whether losing a gene causes a growth defect, reflecting on the fitness of that gene. Well, instead of screening the effect of losing only one gene, I measured the fitness of the combined effect of losing 2 genes at once. And I did that using a combination of CRISPR interference and high throughput sequencing. This resulted in a genome-wide genetic interaction network describing the importance for growth of a very large number of combinations of genes. By analyzing this network, I was able to find combinations of genes which have a synthetic lethal interaction, meaning that when both genes are deleted, a copied stop in growth occurs. A synthetic lethal interaction can be used to identify genes having redundant functions, which ensure correct division and growth of the pneumococcus. In addition, I also identified some genes that positively interact, meaning, for example, that gene A removed the requirement of gene B. Identifying genes that remove the requirement of other genes is crucial. Imagine if you are targeting a protein with a common antibiotic and a simple mutation in a gene completely suppresses the effect of the antibiotic. Thereby, I sought to dismantle some of the mysteries around the cell division of the pneumococcus by identifying new genes in order to develop novel therapies to fight this opportunistic human pathogen.
So thank you, Julia. With that, I can pass the word to our Jerry. If they want to ask questions, the participants, they can unmute themselves and ask.
Hi, I'm Katarina. Hi. First of all, congratulations to all of you. It was great to see the presentations. And I have one question, which actually either Alfonso or Julien, you can try to answer because it's very simple streptococcus question. So why am I always getting a terrible streptococcus infection and I'm 1 week knocked out and my son who gives it to me, he's 2 years old, has nothing? Is this something that has to do with my immunity or is it something with also the balance of other bacteria? Why could this be so?
I think Julien can go ahead if you want.
So I think I will answer by, yeah, there is a lot of factors that decide whether it goes pathogenic, and this ensure this englobe actually, as you said, most of the other bacteria that reside also in the nasopharynx. And as I said during my presentation, it's also Streptococcus pneumoniae is also very directed to immunodeficient people, elderly and children. The fact that maybe your child does not have a a disease afterwards, I will highly, I think most of the bacteria that are around have a high impact, and I also think that, I don't know if you want to add something, Afonso, but yeah, other than the interaction with other bacteria and just the work of your immune system, maybe it was depleted because of other reasons. I don't see any
lack
other
of
reasons.
sleep. Yeah. Okay. Alfonso, do you want to add something or I go ahead with the other question?
I think Julien pretty much explained it well. I think a lot of the reasons are still unknown, but people think it's related with basically the flora that's there at the moment and the immunity. Yeah.
Thank you. And I also have a question for Elodie. So I think it's really amazing that you are trying to find the bias and that the bias is really there. But are there any effective strategies that we can use to fight this bias so that we try to eliminate it?
Well, something that works usually in the medical field is guidelines. We love guidelines. So there has been guidelines actually edited by the College of Cardiology, American Cardiology, that says, like we say in French, like black on white, you should treat men and women the same when facing ischemic heart events. So we try to have something to overpass this problem. But the thing is, this is for the biological part of the problem. There are all this other problem that is psycho-problematic, It's more like we think that it's a man problem, so we treat it as a man problem, even though the evidence and the guideline says, no, you should do exactly the same for both. So this part of the problem is much more anchor and much more problematic because we can't put guidelines to change our mentality. So I think it's slowly with the education of new, I don't know, physicians, new health professional, that we're gonna change slowly things. But I have personally hope, because in my work, I actually have people not knowing what I do as a thesis that come and say, oh, you should be careful because, you know, women sometimes they have the same thing and we should let them less well. So crossed fingers that slowly mentality are changing.
Okay, thank you.
Welcome.
Okay, can I ask a question?
Of course, Dimitri, go on.
Yeah, so hi everyone, and congratulations on your presentations. It's great to hear well-prepared talks, and yeah, congrats. I have a question for Afonso. So I understand from your presentation, so
Yeah.
you discovered the genes which are important for survival of Streptococcus in a competitive environment with Staphylococcus. Is this correct?
Correct, yes.
Yeah, so my question is, can you suggest a way your findings can be translated, for example, to develop a treatment or something? Is there a, let's say, translational application, or maybe in the future, like this can be based on your findings?
I mean, at the moment, it's a bit, could be a far-fetched answer, but you could envision it as, for example, Staphylococcus aureus infection treatment in a way, by using Streptococcus, for example, or even with other bacteria. So basically try to use some kind of mechanism that Streptococcus is using to fight other bacteria, use that to fight those bacteria. But instead of Streptococcus doing it, we do it. So that would be, I guess, the most medical translation application.
Okay, thanks. And another question, just maybe a little bit deeper, is that I was just wondering how exactly having this gene helps the bacteria to survive. It's just like it is able to get more nutrients, or it divides faster, or how exactly?
We are still in the early stages of this. At the moment, I was mentioning this gene that seems to be a protease, so it degrades proteins. At the moment, we think it's just related with basically degrading the other invading bacteria in a way. So basically by starting to digest them, basically.
Thanks.
Yeah.
Are there any questions from the audience that would like to be asked? Nobody has something they want to solve? So if there are no questions from the audience, I want to thank the first three presenters, and we're gonna have a 3 minutes break so we can exchange people within the rooms. And we'll see you later in a moment. Please stay here. Thank you, guys. Okay, so our first participant of the second section of the live on-site presenters is here. So I'm gonna need to move and no. So welcome, Morgan. Dimitra, are you ready to start counting?
Yes, I am, but I also suggest that Morgan can use the timer so that she also sees the time that she has. It's one next to the speaker, or if she wants or not. It's an option.
Do you see it?
It's okay like this.
Okay, yes. So go.
Sorry, my slide.
It's here. So. Okay.
Can we start the time again? It's very valuable time.
Yeah. So go.
I decided to study the human immune system because I find it fascinating how our body can defend us from the exterior threats. What is even more interesting is when our own immune system fails us and starts attacking ourselves. This is autoimmunity. This is systemic lupus erythematosus. In lupus, almost every cell of the immune system is not doing what it is supposed to do. We have the generation of antibodies against self-antigens, against our own DNA, and this leads to massive multi-organ damage. Today, we do not have a cure for this disease. The main reason for this is that we don't understand what causes this disease. So in our study, we decided to focus on natural killer cells. Physiologically speaking, these cells are the first line of defense against cellular infections. In lupus, NK cells are reduced in number as well as in function. They cannot kill cells as they should, and they cannot communicate with other cells as they should. An important thing to know about natural killer cells is that their exterior part, their extracellular phenotype, is actually tightly linked to their function. Imagine, for instance, a soldier going into battle with or without his armor on. So we decided to look at this extracellular phenotype. And here we found two main differences between the lupus NK cells and the healthy counterpart. Actually, we have CD38, which is a receptor that was massively upregulated, much like a soldier going into battle, having way too much armor on. And SLAM7, which was not properly upregulated when the cells are activated. So like a soldier going into battle, but not being able to draw his sword. So we wanted to try to engage these receptors by using monoclonal antibodies that are currently being used to treat cancer. Elotuzumab targeting SLAM7 and daratumumab targeting CD38. And here we found that we can reactivate our immune cells, our NK cells, and not only are they reactivated, but they go and kill the cells that produce autoantibodies. The study suggests that we can restore the function of lupus NK cells by using elotuzumab or daratumumab, and that this will possibly be a new therapeutic approach to treat this disease. And not only lupus, but any other autoimmune disease that is based on the presence of autoantibodies. This is my time. Thank you very much.
Thank you, Morgan. And with that, we can pass to the next participant. Afonso, we will try to improve the sound. Yes, maybe are they too far? Thanks for notifying us. So I guess this is the best we can do. So please be loud.
Okay, we'll try.
So I'm gonna go back to yours. So welcome, Carlos, and I'm setting up your timer.
All right, thanks.
Dimitra, are you ready?
Yes, but one is enough, I guess.
Okay, it's just that he sees. Okay, so do you see it from there? If you prefer this thing. Okay, so go.
So science communicator likes to say that there are more neural connections than stars in the universe. Well, this could seem like a really big number when we go to understand what is human consciousness, when we try to understand what is cognitive, cognition, etc. But the thing is, like, a reality is always way more complex than what we can think on that. And the point of my PhD is all about to put new players into the game. So when you think of the brain, you only maybe think of neurons and these kind of connections. But the truth is, like, there are many other cells that are having important roles in not only how the metabolism works in the brain, but also how the brain computes information. One of those kind of cells is the one that I'm studying, and it's called astrocytes. As you can see in the left, this seems like a constellation full of cells But if we go closer, in fact, those cells contains like a really busy daily life. They go to the blood vessels, they take the glucose, and they prepare the food, let's say, for the neurons to keep working. But not only that, they also wrap around synapses and work in running, so neurons can do their work in a fluent way. The thing is, like, these guys are really complicated to study because, as you can see, they look like a really intermingled. So it's like a really complex morphology, the ones that they have. Our goal here is to try to understand how they can do all those tasks and how they, in fact, codify information that they get from the neurons and from the blood vessels. In this case, what they do is basically they tend to create wave of calcium inside their own morphology. And this wave of calcium can go from the tiniest part of the cell to the whole network of astrocytes, because yes, those guys, the same as neurons, they are well connected, creating another layer of computation in the brain. In order to do that, the interesting thing here is like you need to merge different kinds of techniques. You need to use the photon microscopy, but you need to go to behavior, you need to go use omics, and many other kind of state-of-the-art experimental techniques. And furthermore, you need to analyze that. My PhD is about to try to join together all that information and condense all the results into one mathematical model that explains how those tiny cells can in fact form this network and can make the brain work better. This is quite important, not only because we want to understand the brain, but also this can help to create new artificial neural network in which we can include those people in order to make them more efficient. And that's all. Thanks so much.
Thank you, Carlos. And with that, we pass to the next participant. That is Yancy. Did I say it correctly? Okay. Welcome, Yancy. Do you want the timer here? Okay. So you have your X there, so that you're catched by the camera. Great. So welcome. And when I say go, you go.
Hello, my name is Vanessa. Today, I would like to share a story about Zika virus with you. So what is Zika disease? People may get a Zika infection by mosquito bite. Fortunately, most of patients won't feel anything when they get a Zika virus infection. However, the infected pregnant woman may have this problem for their babies. It's called microcephaly. So what is the microcephaly? The mechanism for the microcephaly is still unclear. So here we would like to study it in vitro. So today, I would like to introduce the Zika virus mechanism for you. Normally, the mechanism for the Zika infection is like this picture. Zika enter to the host cell by receptor binding, and then they will release their RNA genome by membrane fusion. A new Zika virus will be assembled at ER lumen. At this stage, it's still an immature form. After the conformational change at Golgi, the immature form will become mature form. Finally, the mature virus will be released. In our study, we would like to reveal the replication cycle for the Zika virus because the mechanism is still unknown. There is no drug and no vaccine for the Zika virus disease. So here we would like to reveal the replication cycle for Zika virus by electron microscope. This is one of our results by neuron cells. At 24 hours post-infection, we can see there were some virus in my picture, and those virus particles are located at ER lumen. In addition, you also can see some cytoskeleton around those virus particles. In the future, we would like to make a 3D reconstruction model in Zika virus infected cells. We hope our study can give more information for Zika virus disease therapy. So this is my presentation. Thanks for listening.
Thank you very much for presenting the result. And with that, we go to our last participant, Elena. The stage is yours. Wait, I'll give you the timer. So, go.
Pain is the primary reason that people seek medical attention, and rightly so. Pain is an excellent sensory system that warns you of actual or potential damage to your body. Like smoke that triggers the fire alarm, strong heat, cold, pressure, and harmful chemicals will activate your pain-sensing nerves called nociceptors. However, for some people, the system malfunctions and a false alarm is triggered. And sometimes it can go on and on and on. A staggering 10% of the common population suffers from neuropathic pain. And many of these people develop it after nerve injury due to trauma or surgeries. Pain lasts much longer after the wounds have healed, and it is notoriously difficult to treat with medication. So how can there be smoke without a fire? What triggers the false alarm, and how can we mitigate the malfunction? In the lab, we used a mouse model of neuropathic pain that displays the same chronic pain symptoms as human patients. They have an increased sensitivity to heat and cold, light touch can become painful, and even spontaneous activity can arise. In this model, we partially damage the sciatic nerve of the leg, which means that we have a mix of injured and non-injured nociceptors in the nerve. I collect those neurons and measure their excitability, or how easy it is to activate them. In fact, injured neurons are activated spontaneously, but they poorly respond to electrical stimulation. In fact, since they have been severed from the tissue that they were sensing, they will no longer respond to the external stimulation. Instead, it is the non-injured neurons that become hypersensitive. They turn up their volume by increasing their firing frequency. We hypothesize that the injured neurons communicate with the non-injured ones via inflammatory mechanisms to trigger their hypersensitivity. In a collaborative project, we want to implement a soft electronic implant around the damaged nerve to silence it. We use gene therapy principles to deliver the gene of a light-sensing ion channel that will regulate the excitability of the nerve in response to the light emitted from the implant. We hope to silence the spontaneous activity and dial down the volume of the hypersensitive nociceptors. This type of treatment could clear the smoke and hopefully lead to a brighter future for many patients.
So thank you, Elena. With that, I would like to thank our participants and ask you to stay here if they have questions and if we have the rest of the participants outside the room. Yes, please. So does somebody from the audience and the jury have questions for the participants? We can call them to come back. I see you waving, Dimitri, but you're muted.
The participants can also answer from here.
Okay, okay. So they're in the other room, they can ask.
Yes, if you could maybe stop sharing or share the first slide.
Yes, sorry.
I think you can answer from here, right?
You can even come here. I can see. So if we did, they're here. Whoever you can call, they can come and they can answer.
Yes, I saw Dimitri was waving, but I don't see him like.
Yes, so I'm ready to ask a question, if I may.
If you can tell us who you direct the question to, so they can come.
Yeah, I would like to ask Yelena.
Yes, hello.
Hi. Great presentation. Thank you very much. Seems very interesting. I really know nothing about neuroscience. And I had two questions. So if you could just briefly explain why some people have higher tolerance to pain than the others, and also like can be trained. And then a little bit of a sci-fi question, if there is a way to make someone not to feel pain at all. And
yeah,
what do you think?
So for the first question, in terms of difference in pain tolerance, that there's many factors like genetics, and also there's sex differences in terms of tolerance to pain. This would have to be due to partially due to the number of receptors that are expressed to sense the different types of either heat or cold and things like that. And then in terms of training, either you have like this epigenetic regulation or like long-term changes in these expressions, or it can also be changed higher up, like in the brain, how you perceive. Not only does the nociceptors that are in the periphery important, but also your mind can kind of trick you into thinking it's less painful or more painful than it is. And in terms of completely silencing pain, it's not really good because you want to, there's patients who don't feel pain at all, and it's a hard life because you tend to harm yourself quite easily without realizing. So it's not something you would want to silence the excess of pain, but not completely remove the sensation. I hope this answers.
Yeah, thank you.
Thank you.
I don't have any other questions, so if someone else wants to go.
Someone else from the jury, Professor Regula. Someone else from the audience that wants to ask a question? Then, if nobody else wants to ask a question, I would suggest that we go for a 10-minute break and we come back, like 13 minutes break, we come back at 5, so that the committee will have the time to judge and come up with the winners of the competition.
Okay.
So thank you, and it's hello everyone, and thank you for staying live with us. We are back with the results. So if I can ask the participants to move closer to the room, please, and we can ask the ones who want to come inside later. And yes, and hello, you're all back. Great, thank you. So for the moment, I'm gonna, when the participants are here, I'm gonna...
They're on the way.
Okay, I'm gonna speak to the committee.
Yes, just there. And please keep distances.
Or maybe, because the jury will communicate feedback, general 2-3 words each of them, and general feedback to all the participants, and then we will proceed to announce. And Dimitra, you can come back to the room as well. So, which one of the jury wants to speak first while they're coming?
I can, maybe.
So if you guys can keep your distances somehow. Yes, and so now that everybody is here, Professor Roland, you want to share your thoughts and feedback with the committee?
Okay, hello everybody. I am Alexandre Roulin. I just remove a little bit so that you can see me. I'm from the Department of Ecology and Evolution. And why I'm here? Because I'm interested in communication more and more, I have to say. At your stage, I was not so interested. That's, when I see you giving this talk, I'm quite impressed. And I think that you are very courageous. This is really remarkable to me that you try to do this exercise. Really, it's very good. And I have to say that it was very difficult to say that this one is better than the other, because you were all very similar in the way you presented this. I think that if there is one winner, it really means that the others should not think, oh, I was not so, no. You were really all more or less the same quality. Thus, it means that you should continue in this direction and try to get some feedbacks from others. That's something I always wanted to get feedbacks, by the way. I never got feedbacks, never. Now, during my whole career, and you know what? When you were giving these talks, I directly wrote an email to my team, and I said, guys, I'm looking at these guys giving the talks, wow. And I think if I can bring you something, maybe we should talk about what they did today, and to try to let you realize the importance of what you do in your PhD, and to try to make something more general, why this could be interesting for the grandmother you meet in the middle of Lausanne, why she should be interested in what you do. Okay, having said this, I would like to say, because the point is to learn something, and I learn myself a lot, I have to say, that's why I wrote to my team. The first thing is that this is very embarrassing to start to give some specific comments. That's what I suggest. If you want, you can contact me. We can have a Skype, Zoom, whatever, and we can start to talk about what you did, what I remember, by the way, and to give more precise, not really advice, because I want to tell you that I know better than you, but what I felt. And that's your choice. You can contact me, and the others the same. Okay, now if I would give some general impression. First of all, I would like to know whether you received advice from somebody, whether your boss was implicated in your talk, and whether you think, okay, I see some people say no, because they don't care, I think. That's the drama. Oh my God. All of you did not receive any feedback, advice, nothing. Voila. You know what I wrote? I tell you. I wrote, I have a colleague of mine, she's a psychologist. You know what I wrote when you were should we écrire? You understand French? No. All? No, okay, I translate. I said, I've understood something today. The university, and I'm sorry, I'm not criticizing you, but I realized something because you did not receive feedbacks from your boss. I realized that the university invests money for communication, to communicate what the researchers are doing, but they don't invest money to teach people to communicate. And this is the drama, I think. Your own professors, they don't care about whether you how to communicate, because themselves, they don't know how to do it.
But it's very nice to have direct link with the decanate here to get some money on communication.
Yeah, exactly. This is, and I wanted to talk to Manuel Pascual because he's involved in communication. That's what I realized. Wow, my God, we invest a lot to communicate our results. There's a journalist doing the job, but that's you should be able to communicate in the first place. And we don't. And we don't invest this money. Okay, this was just to tell that. Some general comments, that's not your fault, that's the fault of the system, and this is why you are so courageous to do it by yourself, and very impressed. Okay, generally speaking, because I will finish, I think the first one, it was a bit too specialized. You need to have a biological background to understand what you do, the first thing. All of you, all. The second one, some people had the slide, and from my point of view, I don't say that I have the truth, but my feeling is that most slides were quite complex, and don't show something you don't explain. Maybe just one thing, say, wow, my God, oh, this is beautiful. You want to touch the heart of the people. And this is a third thing. You have to touch the heart of the people. You have to make the people your friends. They love you, they want to love you. Thus you need to find a way to, not to show off, but to be nice, you know, say, wow, this guy, I want to have a drink with this guy just afterwards. And maybe there was a last point, a bit specialized. I think generally speaking, maybe we don't agree, maybe Katarina and Dimitri, I don't know, I'm not sure. But I think you tell too many things. This is only 3 minutes, but still, there was a lot of information. And the very last point, I think this is the most important one, is that you have to go out of what you do. Why this is so interesting to anybody? What is the connection between what you do and the connection with the psychologist, the social scientist, I don't know, everybody. What connects your PhD with the daily life of anybody on the planet? And this is something which is very difficult, and it brings the concept of interdisciplinarity. And that's why I wanted to talk with my team, to ask them why you do this, why do you think this is so important? And I had this discussion yesterday evening with my PhD. I showed him the book of the biography of Steve Jobs, and I took some quotes. I said, look what he said. He said to the guy of Pepsi, for instance, he said, you know this story with the Pepsi guy? Because he wanted a communication guy to sell the Mac. And the very last thing he told him, he said, okay, guy, do you want to sell your sugar liquid for the rest of your life, or you want to change the world? And the guy said, wow. And he decided to join Steve Jobs because of this very sentence. And I think this is the conclusion of what I wanted to say.
Great and inspirational, I hope, for everybody. So thank you, Professor Roland. And I give the speech to Dimitri first. You want to add comments?
Sorry?
You want to add comments, feedback, Dimitri?
Yes. So yes, I just want to first congratulate everybody for the efforts and really interesting presentations. Some of critical feedback can be exactly as was stated already, that some things, let's say, were too specific. And I'm not talking about the slides, I'm talking about the presentation. And to me, even though we are all scientists here, but even though some things are a bit difficult to understand. So for non-scientists, I think it would be even harder, especially the usage of terminology and scientific terminology, because in our minds, so we use a lot of words daily, which, things like upregulated or expression of genes, which for us are okay, but in fact, people outside of academia don't know what they mean. So, but it's a challenging task. It's a challenging task to translate it to the simple terms, but keeping also the meaning and the importance. So I can say that some of you did better than the others, but I guess all of you are just in the beginning of this journey in science communication. So the next time you participate in this or similar competitions, you'll do better and you will learn from this. That's the main criticism that I have. Other than that, it's really difficult to compare presentations that were given via Zoom versus the ones that were on stage, because obviously when you're standing and presenting, you're more engaging, you use more gestures, and you feel maybe more comfortable than talking to the audience, which even if there are a few people, then talking to a screen while being alone at home. So for me, I didn't take this into account. But yeah, so anyway, congratulations, everybody. was very interesting, and good job.
Thank you, Dimitri. And then we pass to Katarina.
Hi. So it's hard to add something to what was said already, but congrats to all of you guys. And really, if there is something I would really like to encourage you to continue with this, with science communication, just go ahead, do competitions, try to do presentations, even for your colleagues, simplify. And that's the key really in science communication, I find in this era, it is really crucial. We need to make, as scientists, we really need to make people understand what we do and in a simple way. So just to touch upon what was said by Professor Roulin, like don't only talk to your bosses, talk to your friends, talk to people who are not at all in science. And if they understand, I mean, it's a great preparation for a public defense if you manage to do one. I didn't, but I would love to, because it's a great way. This is the point I see, because your colleagues, they know what you do. People at conferences, they know what you do. So just go and simplify and make the main message pass. I would say that's the key. And yeah, I don't think regarding criticism, all was said. I think all was said. I don't have anything else to add. Just congratulations and courage, and go on with this.
Great, thank you very much. So with that, I pass first to announce the winners. So if you, Dimitra, want to get ready with the presents. So we have 2 presents for the first winner and the runner-up, which are, what do you expect in Switzerland?
Chocolate.
Yeah, of course, it's chocolate. Okay, so I'm starting from the runner-up. So runner-up is Carlos. Can you have a second, Carlos? Yeah, I turned the camera so you could go there. Okay, and the first one is Elena. But congratulations to all of you. As you might have noticed, it was difficult to select the people. And for that, we also want to thank our jury for joining us. For that, we have got something else.
Wow, bottle of wine.
Yes, of course.
It was worth coming. Thank you.
For you guys on Zoom, we have to arrange somehow that you get your present.
I have it now.
If you want to see.
I have. Okay.
Thank you for joining us.
I will steal yours.
I want to also, at this point, I want to thank both Nassim and Dimitra. Where are you? Since I'm trying to catch you here. They're the organizers from ADAS that helped this organization take place. And they were running and stressing with the organization to manage. And ADAS for supporting us. And then I also want to thank every participant for participating. We will send you your participation certificates. And also, yes. We will sign it so that you can get your credit from the doctoral school. So then you will receive this by email. So contact us and we will send them to you by email. So thank you very much. It was very nice, and thank you for supporting this event for the first time in our university this year. And we'll have to see you again live one day. So thank you for joining.
Okay, bye.